Hello everyone, this is Akshay over here and welcome to the next continuation part on excreted products and their elimination. So basically in this particular tutorial we will be concentrating on urine formation and urine concentration mechanism, and later part is as usual with the disorders of the excretory system and the regulation of kidney function. So let's get started. So urine formation. The urine formation process mainly includes three steps, that is glomerular ultrafiltration, next is reabsorption, and the next one is the secretion. So from the previous video, we have discussed that the ultrafiltration process that is mainly vital for the urine formation, and ultrafiltration is mainly a pressure dependent process, right? And how does this pressure is actually generated in the glomerulus? It is mainly because of difference in the diameter of afferent arteriole and the efferent arteriole. So although the blood enters easily into the glomerulus by the afferent arteriole, while exiting from the glomerulus through the efferent arteriole, it is noticed that diameter of this blood vessel, that is efferent arteriole, is less, slightly less than the afferent arteriole. As a result, a pressure gradient is generated inside the glomerulus, which ultimately favors the ultrafiltration process, and the primary urine or the filtrate is collected in the Bowman's capsule. So now around 1100 to 1200 blood ml per minute is actually filtered, and the amount of filter that is actually collected in the kidneys per minute is called as the glomerular filtration rate, or GFR. So the standard value for GFR is 125 ml per minute or 180 liters. So in a normal healthy human, you can see that the urine that is released per day is 1.5 liters. So the next process that is in the urine formation is the reabsorption and secretion. So remember, reabsorption here mainly refers to the absorption of contents from the primary urine from renal tubules back to the blood. But whereas secretion mainly refers to secreting or release of the products that are to be excreted. That is, the movement of molecules or the substances will be from a blood vessel, that is the vasa recta, towards the renal tubules of nephron. So around 70 to 80% of the electrolytes, that is the sodium, chlorine, and water, are actually reabsorbed at the proximal convoluted tubule, which can be seen in this particular diagram, right? So this diagram actually shows which part of the nephron is permeable to what all electrolytes and other substances. But whereas the proximal convoluted tubule secretes hydrogen, ammonia, and potassium ions, and you can also see that even bicarbonate ion is also reabsorbed back. However, at the loop of Henle, the reabsorption is very much minimum, but mainly plays a role in the urine concentration process, which we will be looking in shortly. So in this diagram, you can see that after the filtration is actually collected in the Bowman's capsule, you can see that the movement of molecules such as the bicarbonate ion, sodium, chlorine, and water and other nutrients is from the proximal convoluted tubule towards the interstitium or the blood vessels. But whereas you can also see that hydrogen ion and ammonia from the blood vessel is actually given back or secreted back to the proximal convoluted tubule, right? So from the proximal convoluted tubule, you can see the loop of Henle, that is the descending limb of loop of Henle, and the ascending limb of loop of Henle, right? So you can see that the descending limb of loop of Henle is only permeable to water. Remember, it is only water that can exit from the descending limb of loop of Henle, right? Now you can see that this loop of Henle again takes a U-turn and starts to move from the medulla towards the cortex, right? So you can see that the thick segment of ascending limb of loop of Henle is mainly permeable to the solutes, that is the sodium and the chlorine. Collectively they have given here as the salt, that is the NaCl, right? So from the ascending limb, you can see the next structure here we can find is the DCT, that is the distal convoluted tubule. You can see that there is reabsorption of NaCl. What do we mean by reabsorption of NaCl? That is movement of NaCl from the DCT towards the blood vessel. Okay, it is reabsorbed back into the blood vessel, and water is also reabsorbed, and finally bicarbonate ion is also reabsorbed. But whereas potassium and the hydrogen ion is again secreted back from the blood vessel to the distal convoluted tubules. right? So from the distal convoluted tubule, the next structure is the collecting duct, which is mainly permeable to water and the urea. Urea is again reabsorbed, and the main reason for the reabsorption of urea, remember, not completely urea is reabsorbed, only a minute amount is reabsorbed, which is important for maintenance of osmolarity. So here you can see that from this diagram, the glomerulus, the Bowman's capsule, PCT and DCT, that is the proximal convoluted tubule and the distal convoluted tubule, are the only structures present only in the cortex. But whereas it is the Henle's loop that enters from cortex towards the medulla, right? So this is an important observation that we have to always remember. So finally, reabsorption is mainly uptake of nutrients from the renal tubule, but whereas secretion is secreting or giving back the unwanted substances back from blood towards the renal tubule, right? So this is the urine formation. Now let us look into the next part, that is the mechanism of urine concentration. So before we directly take a plunge into the concept of urine concentration mechanism, let us look into the some of the factors that are very much important for us to remember in the urine concentration mechanism. The first one is the countercurrent flow, and the next one is the differences in the osmolarity, right? That is mainly a gradient of osmolarity is formed from the renal cortex towards the renal medulla. And finally, the structural differences in the loop of Henle, that is mainly with respect to permeability. So now let us look into the first point, that is the countercurrent flow. So the countercurrent flow is mainly the flow of a fluid in the opposite direction. Okay, and this opposite movement of fluid is mainly observed both in the vasa recta and also the loop of Henle of the nephron, right? That is the descending limb and the ascending limb. So here the arrow mark shows that the fluid which is present mainly in the descending limb is moving from cortex towards the inner medulla, and now the same fluid in the nephron takes a U-turn and starts to move in the ascending limb from inner medulla towards the cortex. So here you can see that the fluid is actually moving in opposite direction, and this is what actually we refer to the countercurrent flow. The same is applicable even in the case of vasa recta, that the blood actually flows from the cortex towards the inner medulla and then takes a U-turn and starts to move from inner medulla towards the outermost cortex. The next point we are seeing here is the differences in the osmolarity. So what do we mean by the differences in the osmolarity or the formation of osmotic gradient? You can see that the cortex is having an osmolarity of 300 milliosmoles per liter, right? But as you move from cortex towards the outer medulla, you can see that the osmolarity is around 600 milliosmoles per liter. But in the innermost medulla, you can see that the concentration is mainly 900 milliosmoles per liter. And finally, the deepest part of the medulla will be having osmolarity around 1200 milliosmoles per liter, right? So now, what is the reason behind such a gradient formation, right? From 300 in the outermost cortex towards the innermost medulla being 1200 milliosmoles per liter, right? So what is the reason behind it? It is mainly because of solutes which is mainly present in the interstitial fluid. So what is interstitial fluid? Is the fluid which is mainly present between the cells of the tissue. It is referred as interstitial fluid, right? So this interstitial fluid is mainly rich in the solutes. As a result, you can see that there is formation of an osmotic gradient from renal cortex towards the inner medulla. So the last point that we had discussed is the structural differences in the Henle's loop, that is mainly with respect to the permeability. So you can clearly see over here that the descending limb of Henle of nephron, only the water is leaving the Henle's loop, right? But whereas from the ascending limb, you can see that only the salt or the solutes such as the sodium and chlorine can leave, right? So now this clearly illustrates that the descending limb is only permeable to water but impermeable to the salts or solutes. But whereas the ascending limb is completely permeable to only salts, but whereas it is impermeable to water. This differences in the descending and ascending limb is very much important for the urine concentration mechanism, right? So as we have discussed all the 3 points, that is the counter current flow mechanism and also the osmotic gradient formation and permeability differences in the loop of Henle, that is ascending and descending limb. Now let us look into how actually urine concentration is done. As the primary filtrate is actually collected in the Bowman's capsule, this primary filtrate starts to move from the PCT, that is the proximal convoluted tubule, and enters into the descending limb of loop of Henle. So as this fluid which is present in the descending limb starts to travel from the cortex towards the inner part of the medulla, during this travel distance, during this travel time, water actually exits from the descending limb. So now, of course, that descending limb is more permeable to water. But why does the water actually leaves the descending limb in the first place? That is mainly because of a gradient of solutes or osmotic gradient in the interstitial fluid that has been already formed, right? So it is mainly because of the solute concentration gradient or osmotic gradient that has been formed in the interstitial fluid. As a result, the water from the descending limb exits and enters into the interstitium, right? This is the reason why water actually leaves the descending limb. So now what happens is that at the end of the Henle's loop, by the time the primary urine or the filtrate has reached into the Henle's loop, U-turn of the Henle's loop, this filtrate which is present in the loop of the U-turn of the Henle's loop is now completely concentrated because all of the water has been lost from the fluid through the descending limb, right? So when it was beginning its journey from the cortex, it was nearly isotonic in concentration. But whereas the moment it reaches into the inner part of the medulla, the U-turn of the Henle's loop, you can see that the same filtrate is now completely hypertonic. But now the same filtrate, which is now hypertonic, starts to make its journey from medulla towards the cortex. So as the fluid starts to move from inner part of the medulla towards the cortex, this part, as it is more permeable to salts and solutes such as the sodium and chlorine. So now this solution, which is hypertonic, by the moment it reaches to the cortex, it becomes hypotonic. Okay. Now, why does it actually become hypotonic? The reason is while it is traveling through the ascending limb, all the solutes and salt is actually leaving through the ascending limb. So now, from the ascending limb, the filtrate, which is now somewhat hypotonic in osmolarity, starts to travel through the DCT, that is the distal convoluted tubule, right? Now from the DCT, the fluid moves into the collecting duct. From the collecting duct, you can again see that this particular hypotonic solution is again traveling to the inner part of the medulla, right? So now again the water is actually being lost from this particular fluid, right? At the end of the collecting duct, you can see that this fluid, which will be collecting finally hypertonic solution or more concentrated urine, right? Just to summarize, you can see that initially the fluid which is present in the renal tubule is actually isotonic, but by the moment it reaches to the U-turn or the U-turn of the Henle's loop, it becomes hypertonic, and the same fluid as it travels through the ascending limb, and by the time it reaches to the DCT, that is the distal convoluted tubule, it has become hypotonic, and now through the DCT and as it passes through the collecting duct, it becomes again hypertonic in nature, and finally a concentrated urine is actually excreted. So one thing that we people have to remember over here is that the most important factor that is playing a vital role for the urine concentration mechanism is the length of the loop of Henle, right? So remember, as the length of the loop of Henle increases, the time that the fluid that is flowing inside the loop of Henle required to cover the descending limb and ascending limb also increases. So as a result, what happens is that the final product that is actually obtained at the in the renal pelvis by covering the descending limb, ascending limb, and also collecting duct, you can see that more concentrated urine is actually formed, right? So if any animal that is living in a desert, right, one of the adaptation you people see is that it is having the ability to produce a concentrated urine, right? Now this is mainly because the kidneys are having nephrons whose loop of Henle is very much lengthier. Okay, the length of the loop of Henle in such kind of animals is very much long. But whereas in the case of fishes, their kidneys, which are having the nephrons, loop of Henle in those nephrons are having a very short length. As a result, they will be always producing dilute urine. So now how kidney function is actually regulated or actually controlled. So what happens is that when there is any loss of body fluids, you can see that the blood pressure starts to decrease. The decrease in the blood pressure actually activates some of the osmoreceptors. The activation of osmoreceptors in turn gives a stimulus to the hypothalamus that actually causes the release of an hormone called as the ADH or antidiuretic hormone. So when the ADH hormone is released, it actually prevents the diuresis and promotes the reabsorption of water at the distal convoluted tubule, that is the DCT. Remember, the collecting duct and the DCT are actually impermeable to water. It is mainly the ADH hormone that can make the DCT and the collecting duct permeable to water, and hence complete reabsorption of water can be done, right? So now let us look into another mechanism called as the RAS mechanism, renin-angiotensin-aldosterone system or mechanism. Okay, so this is a way more complicated system that you people see for maintaining the proper kidney function. So now what happens is that when there is a decrease in the glomerular flow, right? So as you people know that the blood actually enters into the glomerulus by the afferent arteriole, right? So when the pressure actually decreases in the front arteriole of the glomerulus, you can see that the special kind of cells called as the JG cells are also called as the juxtaglomerular cells. So remember, the JG cells or the juxtaglomerular cells are actually present in an special kind of apparatus which is present in the afferent arteriole called as the JGA or the juxtaglomerular apparatus. The juxtaglomerular apparatus is having these cells, that is the juxtaglomerular cells. Okay, so now the juxtaglomerular cells of the afferent arterioles start to produce an enzyme called as the renin. Okay, remember, do not confuse this renin with the digestive enzyme of the renin which is present in the stomach, right? Both the names are renin, but the spellings are different. RENIN and RENNIN, right? So the RENNIN actually comes in the stomach part, but this renin, that is RENIN, actually comes in the kidney. So now the juxtaglomerular cells actually produces an hormone called as the renin, and this renin is very much important for conversion of an protein called as angiotensinogen to angiotensin 1. So remember, angiotensinogen is actually inactivated form which is produced from the liver. Okay, so it is converted into the active form called as the angiotensin 1. But however, you also see that this is not sufficient. Angiotensin 1 is not sufficient. It is again converted into a more potently active form called as the angiotensin 2, right? So for this, completely different enzyme is required. Renin is not required for conversion of angiotensin 1 to angiotensin 2. It is mediated by another enzyme called as ACE, angiotensin converting enzyme. Now, the angiotensin converting enzyme actually converts the angiotensin 1 to angiotensin 2. Now, this angiotensin 2, you can see that it is having two important functions. It can actually cause the release of aldosterone. It actually causes the release of aldosterone and vasoconstriction of afferent arteriole. So what happens when the aldosterone is actually released from the zona glomerulosa of the cortex of adrenal gland? There is a reabsorption of sodium and secretion of potassium. So now the vasoconstriction of the afferent arteriole is also having an important implication, that is it causes the increase in the blood pressure. So this is mainly the renin angiotensin aldosterone mechanism. So now there is also one more hormone called as the atrial natriuretic factor, which is mainly coming from the heart, also called as the ANF, right? So remember, this hormone is actually having an exactly opposite function of aldosterone, that is secretion of sodium and absorption of potassium, right? and it is also vasodilator. Atrial natriuretic factor is a vasodilator. So this is the main function of atrial natriuretic factor, which is mainly coming from the heart. So now let us look into the next part, that is the micturition. The release of urine is called as the micturition, and actually it is a neural reflex under the control of central nervous system. So what happens is that when the urinary bladder is completely filled and there is a stretching of smooth muscles because of filling of urine. So this actually is a signal to the central nervous system by the stretch receptors which are mainly present in the urinary bladder. Now, this results giving off some motor messages or the responses from the central nervous system. The motor messages from the central nervous system actually reaches to the smooth muscles of the urinary bladder. As a result, there is a contraction of the smooth muscles and also opening of a urethral sphincter. Okay, the urethral valve. What happens? There is a release of urine. So this is the micturition reflex. Now, when we look into the amount of urine that is excreted by a healthy human, is that it is around 1 liter to 1.5 liter every day. And when we look into the property of urine, it is actually a light colored yellow watery fluid, and it is having a pH of 6.0, okay, slightly acidic. So around 25 to 30 gram of urea is actually excreted every day. The presence of glucose is also called as the glycosuria, and ketone bodies is also called as ketonuria. So the presence of glucose or ketone bodies actually indicates that the person may be having diabetes mellitus, right? Remember, presence of glucose or ketone bodies is actually abnormal constituent. It is not a normal constituent in the urine. So now let us look into the some other organs which are mainly involved in excretion, right? So the first organ we can look into here is the lungs, which mainly excretes carbon dioxide. Okay, around 200 ml per minute, you can see that carbon dioxide is actually excreted. Now, the another structure which is involved is also the liver. Liver actually excretes the bile pigments, excess of bile pigments, that is the bilirubin and bilirubirin, and cholesterol degradation and steroid hormones which are also degraded, and finally some of the drugs which are consumed by the person are also degraded by the liver and it is excreted. So finally we can see that even skin is also having two special kind of glands called as the sweat gland and the sebaceous glands. The sweat glands actually produces sweat, and the sweat is containing the NaCl, urea and lactic acid. And finally, the sebaceous glands produces the sebum, and the sebum mainly contains the sterols, hydrocarbons, and the waxes. And the sebum actually acts as a protective coating over the skin. So finally, we will look into some of the disorders of excretory system. So the first disorder we can look into is the uremia, right? So now what happens is that in the case of uremia, there is accumulation of urine in the blood. As a result, there is a body swelling, right? So the best option to overcome the uremia is the dialysis. Dialysis is basically an equipment where from the human body a convenient artery, the blood is drained from this particular convenient artery into the dialysis unit, and the dialysis unit is actually having the fluid which is having almost all the components of that of the blood, but except the metabolic wastes which are present in the blood, but it is absent in the fluid of the dialysis kit. So what happens is that the nitrogenous waste or the metabolic waste exiting from the blood and entering into the dialysis fluid. Later, the blood, which is now purified, enters back to the human body. So this is a brief description of dialysis. The next one we are having is the renal failures, and the only option for the treatment of renal failure is the kidney transplantation, where the kidney is actually taken from a suitable donor. It is transplanted to the patient. Of course, it is a very complex mechanism because there may be also chance of rejection of the kidney in the recipient, that is the patient. We will be looking this particular part in the human health and diseases. Finally, next is the renal calculi, that is a formation of stones or crystals of salts are mainly formed in the kidneys. The last one is the glomerulonephritis, which is mainly inflammation of glomeruli, right? As a result, the filtration capacity of the glomerulus is actually lost. So these are some of the disorders of the excretory system. So we will stop our discussion here only. The urine concentration and urine formation mechanism is somewhat very much important for the whole chapter. You may expect one or two questions in your NEET examination with respect to concept oriented or analytical oriented kind of questions in the NEET examination. But in the case of CET examination, direct questions may be expected. So if you found this particular session very much helpful for you people, then do like this video and share it among your friends. And finally, uh, subscribe to this particular channel, and do not forget to express your opinion in the comment section. Thank you.